We're now at part 3 of this lecture set dealing with these modes of inheritance that relate to sex. And the first this part of this lecture series, we talked about two autosomal modes, sex limited and sex influenced. Part 2, we came back and we took a moment to talk about a couple of models of sex determination. How do we get to be the sexes that we are, at least from a genetic component? And we talked about the protenter mode, which excuse me, is a mode where you have a sex chromosome. You either get 2 copies or no copies, right? And having 2 copies makes you one sex, usually female, and having only one, so an odd number of chromosomes, makes you the other sex. Then we talked about the Ligase mode, which was where you have 2 distinctly different types of sex chromosomes paired up. They will pair, they have enough component that they can do that, but the genetic information on one is not at all what's on the other. So these are the Ligase mode. And we talked about the fact that we had birds having ZZZW, where the males are the homogametic sex. And we talked about things like mammals and humans, which are the XXXY, where the males are the heterogametic sex. So what I thought now was we would come back, introduce the third mode of inheritance that's related to these sex issues. And this is the one that's directly related to these being on a particular sex chromosome. Okay, so we're talking now about moving over to away from the autosomes to loci that are specifically carried on one of these sex chromosomes. Okay, again, this is the whole landric one, as we call it in mammals. The Y is generally fairly small as far as the amount of genetic information. So we're going to basically concentrate on the X chromosomes. But when we say sex linkage, which is what we're about to say in a moment, that can be pretty much any type of expression that we would see. Okay, all right. So sex linkage, it's a mode of inheritance. So it's a mode of inheritance, right? So this is our third mode of inheritance related to sex. This is one, but it involves physically being located on a sex chromosome. So this chromosome is determining whether you're male or female, but also there are thousands of other genes on there, and those genes are being carried with this chromosome that physically determine your sex, right? Now, we've tried to teach you keys. We call them signatures of various modes of inheritance. With dominant recessive, we said you make the cross, and one of the phenotypes will disappear in the F1, it'll reappear in the F2 as a 3-to-1 ratio. That's a signature. In codominance, you'll cross 2 parental true breeders and you'll get something completely different in the F1, but it'll be equal expression of both the other 2. Lethals, we talked about recessive lethals, and the signature or key for that was the appearance of a 2-to-1 ratio, because that told you that you had lost one of the genotypes. So sex linkage has a key itself. It has a signature. And where we're going to see that signature is this. The results from reciprocal crosses will differ. Okay? Now, what this means, a reciprocal cross, is this. If we set up a cross where maybe we have a green male and we cross it with a red female, you go to the F1, the F2, you get an outcome. Okay? Reciprocal means that you reciprocate. Okay? You do something, but you do it slightly different. And what we're going to do differently here is we're going to change the male to red and the female to green. That's all we're going to do. Okay, so we're going to set up the same basic cross, same loci and all that, but we're going to have 2 forms. Now, for Mendel, didn't make a bit of difference. Didn't matter if you got the wrinkled pollen from male plant, or whether you got the wrinkled as an ovum from the female plant. It didn't make any difference. You always got the same outcome. With sex linkage, that's not going to happen. We're going to see that reciprocal crosses will differ. So where did this stuff, reciprocal, come from? Well, it actually came from Thomas Hunt Morgan. Mendel's ideas had been rediscovered about 1900, and people started trying to do experiments, realizing that these Mendelian traits were kind of interesting and that we finally had a model. Hunt was interested in working with fruit flies, and this is actually his lab at Columbia. You can see the fruit hanging here and working with fruit flies. One day he was looking at through his little microscope, which is actually shown right here, looking through his microscope, and he observed what was normally these red-eyed flies, right? They're Hemizygous, as they're called, the typical wild type for the fruit fly, the Drosophila melanogaster, that in his case. And there was an individual that was a male that looked like this. Now remember, we can tell males from females because males have the solid coated abdomen. They tend to be a little smaller than the females, right? So he could easily tell that he had a male, and he saw this male with this white eye, right? And so he was able, actually, I think the first one may have escaped, but he was able to get some males. So he set up a cross, okay? And he set up the cross looking like this. He said, okay, let's take some of these red-eyed females, because that's what he had, right? He didn't have any white-eyed females at the time. Let's cross her with a red male, okay? Excuse me, with a white-eyed male. And when we do that, let's see what we get. Well, sure enough, when he did it, everything was pretty good, right? We're sort of following a Mendelian process here. We've got two different phenotypes. One of them has disappeared, and this red looked exactly like the female that was back up here, all right? Now he went on, as Mendel did, and produced the F2. So he took some of these reds and he crossed them, and when he did, he got a 3-to-1 ratio. He said, oh, wow, this is just a simple little Mendelian trait. I found this really cool mutant I should look at. But as he was digging through the flies that he got, he noticed sort of an odd issue, okay? And that issue was that the red ones, there were both male and female red ones. But when he looked at the white-eyed flies, the only type he could find were males. So initially he thought, I guess maybe I've got a lethal here or something, right? Maybe the white condition is lethal to females. We don't exist. But it was shortly after that he found a white-eyed female. Okay, so he said, no, it's not lethal. They exist. I just hadn't found them yet. But at first he thought, wow. But he did notice that this is kind of odd that you would get male and females would be red, but only males would be white based on this cross. Well, once he had some females that were white-eyed, he did what good scientists would do. He did a reciprocal cross, right? He turned them around. So this time he took a red-eyed wild-type male. So this time the male looked like this, and he found these females that looked like this, and he crossed them. Immediately, he got something that he didn't expect. As soon as the F1 appeared, there were red ones and white ones. Not all red, like Mendelian process, but half red and half white. And interestingly, all the red ones were now females, and all the males were white. Well, this is kind of weird. It looks like whatever the male had, he passed it down to his daughters, and whatever the female had, she passed it down to the sons in some way. And that was really weird. Well, but being a scientist, he had some females, he had some males, even though they were different colored eyes, and he thought, well, I better make my F2 and see what happens, see if I get my 3-to-1 ratio back, see if I can figure that out. When he did, lo and behold, no, he got a 1-to-1 ratio. And in fact, when he looked at these, he found out that there were some red males and females and some white-eyed males and females. Now that's completely different than what he got in the first cross over here. Now this is a classic example of what I mean when I say that sex linkage gives me a different outcome, right? All I did was a reciprocal cross. I just flipped around which sex had which phenotype. Immediately I get a different answer in the F1. One of them looks like a very classic dominant recessive outcome until you look very closely at the sex of the final progeny. And this one doesn't look anything like a normal Mendelian outcome, right? You almost immediately see there's a difference, and certainly see the difference when you carry it to the F2. All right, so let's take a look at what's going on here. All right, here's the cross that we have. Here's what's going on. Now, first off, it's quite true that red eye is dominant over white. Okay, it's a simple dominant system. There's nothing odd about that part of the model. It's a simple dominance model. Okay, now what's not so simple is this. Okay, fruit flies turn out to have a Ligase mode of sex determination. at least for our purposes, that's what we're going to use. Okay. Females get 2 X's, so they get 2 copies of every allele. The males only get 1 copy of each allele. Okay. So in our case, the white-eyed male has a Y, and then he has an X chromosome where this is being carried. Now, if you feel uncomfortable writing like this, a lot of people tend to write it maybe like this, where they take the X's and they indicate the alleles that are carried there, and then the male over here would only have 1 X and carry. Either way you want to write it, that's fine. It doesn't matter. I'm just shorthanding it over here, okay? But it's the same outcome. All right, we have a new term, an important new term. Males are hemizygous. At least in the Ligase system in the XXXY, the males are half are hemizygous, meaning you've got this big X, you've got a locus on there, the Y's here, but it doesn't have any of that genetic information. It's just missing. It's like it's not even there. Okay, so whatever the male gets on the X, they express because there's no counter allele on the Y to do anything. So this is known as being hemizygous. Okay. Being sex linkage is about the only place that I use Punnett squares, because in reality, you're actually kind of thinking of it as a dihybrid cross, right? You've got some chromosomes that are genetically making your sex, male or female, and at the same time, you're carrying along this information from another locus that it doesn't have anything to do with sex determination, but happens to be riding around with the chromosomes that do. So it's in some weird way you can think of it almost as a dihybrid, but be careful, don't go putting, some people for whatever reason put XX and then RR next to it, and that truly makes it a dihybrid, and it really will mess you up. So don't do that. Okay, but if you want to write it like this, that's fine. Okay, all right. So what are we going to get? We're going to make a cross between a homozygous female who's going to pass red to all of her progeny, right? Red's a dominant, so everybody's going to be dominated. Even though the male's passing a white, it's dominated in the females, and then it's not even showing up in the males because the sons of this are getting the maleness from dad, the Y chromosome, as we see there. Okay, so we've got these 2 outcomes down here now, right? And we've got this basic idea that we should have this. So we've got these red males. Now you notice that the females are all going to be heterozygous, but because of dominance, they're going to be red. The males are going to be hemizygous. Okay. Well, now we cross these with these to see what we get, how that makes the F2 come out. So we throw these together. The female now can pass down either red or white. The male is only passing red, all right? Okay, so he's passing that down. So what we end up here with is that we get some homozygous reds, we get some heterozygous reds, and we get some hemizygous reds. Okay, so homozygous, heterozygous, hemizygous. And we get this white hemizygous here, right? Because mom passed down an X carrying white on it because she had one to pass, and dad just gave him a Y. So there's 1, 2, 3 red male individuals, right? So that accounts for our 3 reds, and it also explains why there's no female that's white. We can't get 2 little R's. We can't get 2 X's together that each have the white allele on it. So that's not possible. So we see a 3-to-1 ratio, but obviously it's out of balance relative to the sex of those progeny. All right, so let's try the other one over here. Let's move over and say, all right, what happens when we do the reciprocal cross over here? Well, we think about it, we still haven't changed the dominance issue. Red's still dominant over white, okay? We cross, at this time, a female who's white-eyed and a male who's red-eyed. So think about what we must see. She's got 2 X's. Since white is recessive, she must be carrying 2 little R's, right? She has to be. So her genotype must be little r. You can put them up on the X's if you want, whatever. Okay. The male is hemizygous, so he only needs 1 copy to express. So he's going to be big RY, things like that. Okay. We take those and we produce this F2, and you can see how we produce it, right? We produce it by this little R coming down to everybody, right? And it comes down as part of a heterozygote in the females because dad's giving the other X. So mom's always giving you an X. Dad, because he is the heterogametic sex, he can give you an X carrying red, or he can give you a Y that's not going to play a role here. Mom then turn. Okay. Now see how that does the crisscross part, right? We talked about that. And that's one of the signatures that you will see in sex linkage. Not every sex-linked cross will have crisscrossing, but it is a classic signature of sex linkage. What I mean by that is mom is determining what the sons look like, right? The white mom is making white sons. Dad is determining what the daughters look like because he's passing along a dominant red. So you got a classic crisscross pattern that you will see very often when in problems when you're working sex linkage. Again, it's not every sex link cross will do that, but if you see a crisscross, that's a classic example of a model for being sex linkage. All right, so we've produced a het here for females. We've produced hemizygous males here. We set up our little Punnett square, and we see that we're getting some red passed down here. We're getting some little r's passed over here, so we're going to get some females that are little r, some females that are dominant red over white. We're going to get males then, because of moms, about half of them are going to get red, about half of them are going to get white, and they're just going to get a Y allele from dad no matter what, right, or Y locus. So of course that gives us the opportunity to have a red female, a red male, a white female, homozygous white, and a hemizygous white male, right? If you put those together, that's where we get our 1-to-1 ratio, right? And in that ratio, we see what's different than we saw in cross A. We see that we've got this female and males, right? There's female and male that are red, and females and males that happen to be white. That's our pattern, okay? And just to remind you, this is one of those cases where you do see the crisscross pattern. So always look for that. Okay, so always look for reciprocal. And a lot of times you'll see genetics problems where we'll say, blah, 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 blah. Reciprocal crosses were done and there was no difference. Okay, well, it's not sex linkage. Okay, our reciprocal crosses were done and there was a difference. Well, then it is sex linkage. Or you go off into space and you discover something in creature, and the blue dads always make blue daughters, okay, and the green moms always make green sons or something. That's crisscross. So there's some kind of weird sex linkage going on there, okay. All right, let's try, as we always do, let's go to a few problems here and see. It dominates in males, but it's recessive in females, so I'm good at polled females. So, we have it, right? So the question, and then you got to finish up the problem. What fraction of the